Okay, I'm writing the exam right now, and what it's looking like right now is there will be one data set that you download. I'll put it up on eCampus before the exam so you can download it ahead of time. It'll be a next-gen sequencing data set, an RNA-seq data set, so it's just read counts, but omics data for us. Thousands of rows, a handful of columns. And I'll ask you to do principal components on these data and cluster analysis on these data just as a quick exploratory thing. So that's old stuff. And then do DE-seq2 to do differential expression analysis. Tell me, for example, how many genes are significant at a false discovery rate of 0.1 or something. And then apply a predictive model, one of them that we've talked about. I'll pick an relatively straightforward way to do a predictive model, like naive Bayes, for example. You just type naive Bayes, and you could follow the syntax on one of the existing scripts that we've seen in class. You can download those during the exam. So you can refer back to what we've already seen, like specifically how to do the DE-seq2 and naive Bayes. So it might be naive Bayes, it might be lasso, I'm not sure, but it'll be something that you can just pick code from an existing script to implement it. But apply one, give me a confusion matrix, tell me about how it did. Interspersed in that might be a couple of conceptual questions, like tell me about the difference between the accuracy of your predictive model on training data alone and what you would interpret if you were to apply that to held out test data, like fishing for that distinction that we've talked about. You overfit if you only look at the training data. Held out test data give you a more generalizable estimate. Cross-validation is an option for doing that without test data. So conceptual questions where, write me 3 sentences that shows me your understanding of such and such. Remember that I'll be here 30 minutes early. Local students can be here 30 minutes early and have that extra time to take the exam. Distant students will have access to the exam at the usual time, but you will have an extra 30 minutes to complete the exam. It'll say it on your cover sheet. Are there any questions about the exam at this time? If I asked, I won't ask you to do a bootstrap, like code it up. If I ask a bootstrap question, it'll be a conceptual question. What is the bootstrap good for? Suppose we wanted a standard deviation for our prediction accuracy. Propose a way that we could do that in words. I would resample with replacement, I would redo the model, I would compute the accuracy each time, and compute the standard deviation of those capital B accuracies. All right. So what I was going to do today was talk a little bit about proteomics. The things we'll talk about here aren't specifically restricted to proteomics necessarily. The main unique aspect of these types of data that we're going to talk about is that they come from a different technology. This is not gene expression microarrays where you have these little spots on an array and you get it in a fluorescence intensity to quantify things. They're not read counts where we're doing sequencing and mapping back to a reference genome. It's different. This is a widely used technology for proteomics. If I want to quantify proteins, why would I be interested in proteomics and not genomics or GWAS or something? Why would you pick between one or another of these types of technologies? If you think back to the beginning of the semester when we talked about that central dogma of molecular biology, we said there's the DNA, that's like the reference manual, contains the static information that defines you. There's RNA, which is a copy of the DNA that is an intermediary step between it turning into action through proteins. And the things that actually do stuff in the cell are the proteins. So looking at the DNA, for example, genetic sequence information talks about structural characteristics, mutations and allele frequencies and things like that that are specific to a setting. If you look at the proteins, though, so we've talked about this thing that the DNA doesn't tell you the full story. The DNA sequence doesn't necessarily tell you the full story about why somebody has cancer, because not all of the genes are on at every time. Some of them will be on to different degrees as others. So what it would ultimately manifest as is proteins doing unusual things. Cancer versus normal. Ultimately, there are proteins, and there would be these other RNA things that are active as well, but predominantly proteins that should be killing the cell when it's time to die, for example, and they're not doing it anymore. So people who do proteomics, like these guys, my collaborators, were at Pacific Northwest National Laboratory, and they're a very active research group, large group, and all they do is proteomics. And that's what they argue for when they put in their grants to the National Science Foundation and National Institutes of Health. We really care about proteins because they're the ones that are doing things. They're biased because that's their bread and butter. But proteomics is a valid technology. It's a valid way to investigate the omics of an organism. All right, I'm not going to read through the whole paper, obviously, but this was a summary paper that we put into an applied statistics journal. So it is kind of it's at a good level for us. This is my favorite reference, since I wrote it, for explaining mass spec, in particular liquid chromatography mass spectrometry. What I would like to do is look at pictures. So let's talk about this process. Every omics technology, if you were to look closely enough, has all kinds of steps involved in taking like a biological sample to a matrix of numbers for you to play with. Features in the rows, samples in the columns. Proteomics is no different, but it is distinct in its technology. It's not microarrays, it's not next-gen sequencing. All right, so let's talk about this picture. This is intended to be just a big picture overview of the whole enterprise of mass spec-based proteomics. We want to quantify protein expression levels. Just like when we did gene expression microarrays, we wanted to quantify RNA expression of genes. Now we're looking at the actual proteins. Just because there's X number of RNA copies that code for a protein doesn't necessarily mean you're going to have X copies of that protein, because you might get X copies of that protein and some of them get modified after they're created with post-translational modifications and methylation and things like that. So looking at the proteins themselves is kind of unique. Here's a beaker full of proteins. So we get a rabbit. We're interested in studying the proteomics of rabbits under starvation, maybe, and here's their proteins. The way we turn proteins into numbers. So there's multiple copies of the same protein in there. Some proteins are highly expressed, some are not expressed very much. Some are absent. They're not being turned on right now in this condition. We need numbers to somehow quantify abundance. Proteins themselves are macromolecules. An RNA molecule is very small compared to a protein. Protein are big molecules. They have three-dimensional structure. And if you try some of the techniques that we've been talking about, like a gene expression microarray, they don't work as well because of the size and the properties of proteins. That's why we often use mass spec. There's also gel-based technologies, but mass spec proceeds. All mass spec does is measure ions. Ions are molecules with charge, like an extra electron or an extra proton. When you have ions, if you want to count up, like tabulate the different species or the different ions that are in here in terms of basically their size, that's what mass spectrometry does for us. So mass spectrometry will give us one of these types of pictures. That's a simplification. It would really look more like this. So mass spec data, there's peaks, lots of noisy stuff, and peaks every once in a while. On the horizontal axis is what's called M over Z. That's mass to charge ratio. It's the ratio of mass to charge. When we do the preparation step, mass spec needs ions. So we have to make our proteins, or the pieces of the proteins, be ions. And what that means is you shoot protons at the material as it comes out of a column in liquid form, give it charge. Now it's ready to go into a mass spectrometer. Everything that goes in there has a mass. There's different pieces of proteins called peptides that are big. There's some peptides that are small. Depending on the amino acids that make up these peptides, they have different mass. And theoretically, we know the exact mass of each amino acid. You know the chemical composition of it, so you can just tally up this many carbons, that many hydrogens, and so on. So in theory, a peptide, which is just a string of amino acids connected to each other, has a unique mass. It is this amino acid and that one and this one and that one. If you add up all their masses, there's a theoretical mass. If we had an accurate enough instrument, we could measure the mass and exactly figure out, or at least try to exactly figure out the peptide. What is that peptide? In practice, we don't have that amount of precision. We have peptides, we give them charge, and now where there's two pieces that we're going to play with. There is the mass that we will get information on from the instrument. But what it specifically gives us is the mass divided by its charge state. If there's 2 extra protons, it'll be a +2. If there's 3, it'll be a +3. So if you take all these peptides and just shoot indiscriminately lots of protons at them, some of them will pick up 1, some of them will pick up 2, and so on. In a mass spec proteomics data set, there's all kinds of charge states. There's like a distribution of them. Lots of them are +1, some of them are +2, and so on, up through +8 and so on. What we get out of the instrument, the raw data, is mass to charge ratio. And roughly speaking, here is a peptide. There's a peptide, there's a peptide, there's a peptide, there's a peptide. There may be other peptides hidden in here, but we're not going to be able to pick them out because the signal to noise ratio is too high down there. So roughly speaking, the task is take your peptides, ionize them, put them into the instrument and get these types of pictures. Then figure out this is peptide X, this is peptide Y, like identify. This peptide X is sequence such and such. Just like in next-gen sequencing, we get a bunch of read counts. So I have a read count for a particular sequence of A's, C's, T's, and G's. 522 copies of that read we're seeing. Before we move forward and do like a differential expression analysis, we need to identify that where that read came from. Identify it. That's where we go back and map to the reference genome that allows us to say that's gene X. Proteomics, we have to identify what we have also. All the instrument gives us is mass to charge ratios. But we know, like in this the hypothetical context, that we're studying rabbits. And we know the genome of rabbits. If you know the genome, you know the proteome, hopefully, or ideally, because the genes code for proteins. So if I know the genome, this gene codes for this set of amino acids, call that protein X. When we get these spectral data, the next thing we need to do is compare to a database. Just like with read counts, we compare the sequences to a reference genome, and we compare to a database by, quote, aligning. Here, we need to compare these M over Z values, basically, to a database of rabbit. Rabbit has this genome, has these proteins. In theory, if you ionize and do mass spec to those proteins, you would expect peaks like such and such because of the composition of that protein. So back here in the picture, we take our raw biological material, extract the proteins. I forgot to mention, because of the nature of proteins being big, we break them first before we ionize. Throw an enzyme in there, chop up the proteins at predictable places. So now instead of giant macromolecules, we have lots of little pieces of peptides, short amino acid sequences. Ionize them so we can play mass spec with them, throw them through the mass spectrometer, and we get these spectral patterns. This is what I was talking about on the whiteboard, identification. We have a peak here, we have a peak there, and so on. Here's a database. Here's a database of amino acid sequences that are predicted for the rabbit. If you take rabbit proteins and you pick this enzyme and put it in with them, it is expected to break, they use an enzyme that breaks at a predictable place in between certain amino acids. So if I know the genome, I could predict the possible peptides that I would see. Then you take a spectral picture like this and you basically match it to the best fit in the database. This particular peptide with this sequence, in theory, if you ran it through mass spec, you'd see something maybe like this, because there's a carbon and there's a hydrogen and there's an oxygen and so on. There's a chemical composition of that peptide. When we have an observed spectrum, might look something like that. Won't be perfectly matching the theoretical one, but the pattern. Can you do some kind of comparison to all the patterns in the rabbit database and pick the one that looks most like it? That's what identification does. Give me your spectral data. We run it through a database, and at the end of it, we have that first peak is peptide X. The second peak is peptide Y, and so on. Along with those best matches are confidence scores. Just like when we do sequencing and align back to the genome, there's different qualities of alignment. It's very rare that we have perfect alignment. So we would like to say this read, we think, came from gene X, and I'm this confident in that alignment. We will get similar information from mass spec proteomics. Once you have done that, you're ready to do statistics, because at this point, you have an M by N matrix of numbers. M is the number of peptides now, those are your features, and N is your replicates. What you would be most interested in then is doing inference, like differential expression analysis or prediction, classification. Probably most interested in at the protein level. Peptides are the data that we have. We have data at the peptide level, but we really care about proteins. In next-gen sequencing, we have read count data. I really don't care about reads. I care about the genes they came from. So I'd like to do tests and put p-values and false discovery rates on genes. Here, I'd like to do it on proteins. So this picture over here illustrates the issue of our having data on peptides but being interested in proteins. This picture is an example from a real mass spec data set where a protein is represented here. This particular protein had 6 peptides that were observed in the data, that were identified. 6 different peptides were identified at least once out of our replicate observations. And for example, peptide 1, these dots above peptide 1, those are the actual peak heights for what was identified as that peptide. We have 1, 2, 3, 4, 5, 6, 7, 8 or so replicates in this data set. So those were the numbers for peptide 1. Those were the numbers for peptide 2, peptide 3, and so on. You need some way to roll up the peptide information to the protein information, peptide level to the protein level. I won't go into detail on that, but it's just a necessary step. If you're familiar with Affymetrix gene expression microarrays, it's an analogous thing that you have to do there. Affymetrix gene expression microarrays, it's a microarray, so you have, roughly speaking, these different spots on the array, and each array or each spot corresponds to a gene. But in reality, if you zoom in real close, each spot actually has lots of spots, multiple spots, different probes that all target in slightly different ways the same gene. Affymetrix calls the collection of probes that target the same gene a probe set. That's what we talked about as a probe set early on. So when people do differential expression analysis with an Affymetrix microarray, they do something like this under the hood. That has to be done. You take the collection of numbers on all the probes that go to that gene, you need to turn those into a number somehow, or somehow roll them up to the gene level. Same thing in proteomics. We have peptide level information, but I'd like to do inference on the whole protein. In this case, there's only one comparison group, so we might be interested in what is the estimated protein expression level for this particular protein. What's the estimated expression level, and give me a confidence interval. That's what the shaded region is. So that's a single number, taking all those 6 peptides into account and doing some kind of sensible weighted average to get an overall estimate, and doing some kind of sensible pooled variability estimate to get an overall standard deviation so that I can do a confidence interval. This is not. This picture, the next few pictures zoom in on different aspects of that big picture story. So this is hard to see, and we'll just look at it for a second. You take that first step of sample preparation, you take your rabbit, extract some tissue sample, extract proteins from that. So protein extraction, I don't want lipids, I don't want RNA, I don't want DNA. There's a step called fractionation, which removes really high abundant proteins. That's often done. You know, if you take a blood sample, for example, in humans and quantify proteins, the signal will be drowned out by some proteins that are always there in high abundance. So if I'm interested in more subtle patterns between a disease and healthy, say, we will want to remove those really high abundant proteins. Digestion is the step where you put the enzyme in. You take the proteins and you digest them, you chop them up into smaller pieces. Now we have peptides. This step at the right is how we do two things. Okay, you could take a textbook, hundreds of pages of words. You could print the entire textbook, if you had a good enough printer, on one piece of paper. Just write really, really, really small. You could put all that information on a single piece of paper. But we wouldn't be able to read it because it's just jam-packed with ink. Can't even see it. A similar thing happens here. Here's a rabbit. Here's all its proteins. Digest it. Now we have a big pile of peptides. Lots of peptides. And if we were to run that entire collection of peptides through a mass spec instrument, we'd be liable to just get a huge mess that we can't see anything in. So for the same reason that you wouldn't write a whole textbook on a piece of paper but spread it out over multiple pages, we spread out the peptides. The way that they're spread out is with, in this technology, liquid chromatography. Liquid chromatography is a chemical technique that allows you to spread out molecules in terms of some chemical property, like hydrophobicity. So what we do with our collection of all the peptides is we put them in this little needle-looking thing, it's like a pen or something, and the liquid that has the peptides in it comes into the LC column, and the column has these little beads, and based on how hydrophobic I think you are, you travel through the column faster or slower. So you pour your things into the column, wait a little while for things to traverse, and then it starts dripping out a little bit at a time. The first drop to come out contains peptides that traveled the fastest. So you've basically taken all those peptides and now you have a small collection. So that first drop, shoot some ions at it or shoot some hydrogen at it, protons at it, and make it ions, and that drop, its ions, become the first spectra that we get. Spectrum. On this picture right here, these multiple spectra are due to the fact that we're separating out the whole batch of peptides, and we're going to take a snapshot mass spectrum for every drop that comes out. So the LC column might elute a drop of material every second or something, every couple of seconds. So the instrument, if you go to the sequencing, or not the sequencing center, but the mass spec center on campus, the instrument, you have this huge box that does the mass spec, and over at the side there's a little place where you put your peptides, and then there's a little column, the LC column, and it just starts dripping, scanning, and so on. I don't know. Hours. I think it takes hours. Yes?
When you chop up those big protein molecules, does your M over Z letters change?
They can change. So if you have one peptide or one protein and you break it into, say, 6 peptides, each peptide can accept its own charge. This peptide could be +1, that one could be +2. Their masses will almost certainly be different because you've taken one long sequence of amino acids and broke them into pieces. So here's a small piece, here's a longer piece, and so on. The masses will be different. So yeah, M over Z can all be different.
And the mass spec breaks down the segment inside of the mass spec too.
Okay, yeah, we'll get there.
It's like a fragmentation of the peptide in the instrument.
We'll talk about that. This is intended to mean a separate spectrum for each little collection of elution coming out of the LC column. That's the case. When we do the identification, what some instruments allow you to do, not all of them, is here's a high peak at this place. That one peak is not informative enough usually to do the identification. But what you can do is, like you were saying, The instruments can capture the ions that produced that peak and run them through again. Only. So basically zoom in on their profile. That one peak is like a total M over Z for the whole peptide. If you were to take the ions that are all from the same peptide, they all gave rise to that peak, get everything else out of the instrument for a moment, run those back through the instrument, then you get what is more of a identifiable fingerprint for the peptide. So if we do our mass spec and we have these peaks in what's called tandem mass spectrometry, or MS-MS, so this is called MS-MS in the sense that we did MS and then we picked some of the ions and we did MS again. And you can do it more times. Sometimes they'll call it MS to the nth power, as in we did it 4 times. But what you get is a zoomed in version of that, which will have its own peaks. So there's a peak for the carbons for that peptide and the hydrogens and so on. And it turns out that when you do this, that pattern bottom right is informative enough with modern technology to, with high confidence, identify that peptide. That's pictures for that type of picture are what is in the database that you compare to for identification purposes. Okay. So this summarizes what we were saying. We have our peptides. We spread them out. We separate them with liquid chromatography. As things come out in batches, they are ionized, put into the mass spectrometer, and then we have lots of scans. My one sample, this one rabbit that we're going to run today, we extracted proteins, fractionated and all that. For that one rabbit, I'm going to get a bunch of scans because there's going to be a bunch of material that comes out of here. So here was the first scan. There's the second spectrum, third spectrum, and so on. You can summarize all the data for that one rabbit in actually 3 dimensions. There is the scan number. Was it the first drop or the 14th or whatever? That tells you something, by the way, because that tells you about the chemical characteristic of that peptide. It is informative, the scan number at which it came out. We also have the M over Z values. So the horizontal axis value. Where did it line up? What was its mass to charge? And coming out towards you from the picture would be the actual height of the peak, the number that we would use to quantify. So here's your data. You ran one rabbit and you have that type of data. What do you do with that? This is some detail on information that's contained in this picture, which is a little bit, I don't know, it took me a long time to get my head around it, but here's that picture from the previous slide. And if you zoom in on that circle, let's see, no, not that circle. If you zoom in on this vertical line segment, that vertical line segment is a scan. So scan number 5,338. We had 13,000 scans all told on this one rabbit. This particular scan, if you were to pick that up, remember that the peak heights are coming out towards us. If we were to pick that up and rotate it, we could make this picture. This is the mass spectrum that came out in scan 5338. So we have 13,000 of these left to right going across that picture. If you were to zoom in on this little circle right here, let's zoom in on one little place. In terms of finding the peptides, all we have from the instrument is a bunch of peaks. where are the peptides? To identify the peptides, we use some knowledge of the chemistry. This picture is intended to be zoom in on the big scatter plot to a place where there is a peptide. If there's a peptide, you're going to have data that looks a particular way. A peptide won't necessarily elute at exactly the same time. All the copies of the same peptide won't elute at exactly the same time. There's randomness. There's these beads in the LC column, and some of them will randomly get through faster than others. Even if the average elution time is 3 minutes for a peptide, it'll be 3 minutes, 3 seconds for some. It'll be 2 minutes, 58 seconds for others. So here is all of these dots, all of these peaks came from representatives of the same peptide, different copies of the same peptide. They showed up over a period of time. And they had different M over Z values. They had a profile of M over Z values. So there is an elution profile. There is also an M over Z profile. And what this speaks to, the way you can have different M over Z values is, A, you could have a different charge state. If you have the same mass, but this version is +1 and that version is +2, this one's going to have half of the M over Z. So you can have a predictable collection of M over Z values. There are also ways, like there's heavy hydrogen versus regular hydrogen, and those show up every once in a while just by chance. So there will be other cases where same peptide that happens to have unusual versions of the elements will have slightly different M over Z. All right, I won't go too much more on this, but the big idea is that you get lots of data, all these different scans. Somebody has to code up an algorithm to search through that to find patterns that look like what a peptide would look like. A profile and a stair-step kind of pattern as the charge states change of the same species. And long story short, you can back out of this information to, this is sufficient to identify the peptide, or to say that it is a peptide. This picture speaks to the identification step. So if we have a peptide with this amino acid sequence, say, we're going to observe a mass spectrum or a collection of mass spectra. This is MS, a fingerprint, the tandem mass spec fingerprint of this peptide. So this is the pattern that we would compare to a database. We would have a mass measurement. This MH is called monoisotopic mass, so that's the mass that's most frequently observed for this peptide. A database would contain, for all the different peptides known for rabbits, the theoretical profile, the theoretical fingerprint, the theoretical mass, and the identification step is to compare and match to the best one. So based on this picture, we didn't know the sequence, but we saw these data, we compared to a database, and we identified as this peptide. We may be right or we may be wrong when we identify. Just like when we do next sequencing, we may make sequencing errors, we may misalign to the wrong part of the DNA. We would like to be able to assess how confident we are, or we can be. Let me just say something about the way they do that. It's kind of unique. In the proteomics context, we get out, when we do identification, we have for each peptide, when we looked at that picture of scan number versus M over Z, and we zoomed in and we found a profile that looks like it's a peptide. So we summarize that into a mass. We extract the MSMS fingerprint for that. And then we compare it to the database. So from the database, we will get a match. Let's call it peptide X and a confidence score. Some kind of match quality. These T values are, I don't remember what they are, but there's some kind of match quality. The higher the number, the tighter the fit. If you take, you're going to have piles of those because you have piles of peaks in your data. For each of your putative peptides, you're going to have a confidence score. And in terms of what you would like to do is do your best for every peptide for everything that you think is a peptide, run it through the database to try to identify it. With the understanding that every time you run it through a database, it's going to give you a best match. This is the best we could do, just like with alignment. Just because it gives me an alignment, though, doesn't mean it was a good one. So try your best to identify with entries in the database, but give me some number. These are not p-values, unfortunately. These are just numbers. But what we can do is think statistically about them. So out of our pile of peptides, we identify against a database and get best matches for each one, together with confidence score. If I draw a histogram of the confidence scores, the however many tens of thousands of confidence scores we have, will tend to look like, in practice, a big pile of false identifications and a relatively small pile of true identifications. False identifications will tend to have small confidence scores, and most of the putative matches, if you take a peptide and compare it, I think we get all the scores. Is that right? No, these are the best scores. These are the best scores for each of the things that we think are peptides. But we expect that most of them will be mismatches. That's just the way, that's my understanding of what usually happens. When you make a picture of the confidence scores, you get a big pile of what the chemists will say are false identification and a relatively small pile of true identifications. So out of my huge pile of possible peptides, I think these are the ones that I would like to focus my analysis on because I don't believe those. Just like that big pile might be a bunch of reads that align to the genome with a really poor quality. So I don't want to include them in my data set. I don't want to say row 18 in my Excel spreadsheet is gene BRCA1 when I'm not confident at all that it really is. So in order to extract the high quality identifications, what you can do is look at this picture, and what we might say is that this picture is a mixture distribution. When we talked p-values and false discovery rates, we made a histogram of the p-values, and we said, let's think of that like a mixture. It's a mixture of a bunch of p-values at the left for differentially expressed features, and a flat distribution for the null features. We could do something similar here. Let's assume that this is a mixture of, let's say, two normal distributions. There's a normal distribution for mismatches, and there's a normal distribution for true matches. And there are statistical techniques, off-the-shelf techniques, for solving that problem. Give me a collection of data that you think came from two normal distributions. The first one has some mean, mu1, some standard deviation, sigma1. The second one has some mean mu2, some standard deviation sigma 2. There are bona fide methods for fitting that model, like estimating the two distributions. You can get estimates of mu1, sigma 1, mu2, sigma 2. If you have those estimates, if you fit the model, then you can quantify in a probabilistic way how likely it is that this peptide was correctly identified, roughly speaking, by comparing which of these two distributions it was more likely to have come from. So if peptide X had a confidence score that was right there, maybe, that's much more likely to have come from the smaller distribution than the bigger one. So I'd be confident in saying that's a correct match. Whereas something over here is much less clear, and certainly something over here I would want to get rid of. What often is used in proteomics is a posterior probability of a correct identification. The use of posterior should bring to mind Bayesian methods, like Bayes' rule when we talked classification is getting the posterior probability that you're class A, given your input vector's posterior probability. So this happens before we have our m by n matrix of numbers, because this is going to be what we use to filter out among the thousands and thousands of features that we found, only those that we think are real peptides. Okay, and just briefly, let me say something about where you go from there. After you have done the mass spec runs, you have identified the peptides and done this quality filtering that we just talked about. Now you have an m by n matrix of numbers. I've got an example on eCampus that I'll show you next time. I'll show you next time. The diabetes data, if you want to look at them, or mass spec data. At this point, we have an m by n matrix of numbers. The numbers themselves are peak heights or sometimes areas under the peak that were assigned to a particular peptide. So now we could say here's another thing. If I identify, we're studying rabbits, and I identify a particular collection of peaks as peptide X from the rabbit. Peptide X has a known sequence of amino acids. We know the genome for rabbits, so we know its proteome, which means if it's peptide X, I know what protein that came from, usually. There are some cases, it's actually not that uncommon for there to be redundancies, like this short amino acid sequence shows up in protein X and protein Y. But what we hope for, and usually get is each peptide comes from a known protein. Once we've identified the peptides, we know the protein. So here are these 6 peptides from our rabbit that come from a protein. Tens of thousands of peptides, maybe 100 or a few hundred proteins usually. Here's one of them. And in this case, there's a treatment and control condition. For each of the peptides, I have some circles, which are controls, and I have some X's, which are treatments. And like we did in that first picture, we'd like to combine or roll up these peptides into a single protein level estimate of control mean and a single protein level estimate of treatment mean, together with standard deviations, so we could get a p-value or do a hypothesis test. In this case, estimated mean expression level of this protein in the control group was statistically significantly smaller than that in the treatment group. So this would have been called a differentially expressed protein. One last detail that is important. Mass spec, often we have m by n matrix of numbers, often there's lots of NAs in there. There's lots of missing values. So a peptide, especially if it's a relatively rare protein, low abundance peptide, it may show up in this rabbit and that rabbit, but we just didn't see it in the other rabbits. It may have been there, but it was in the weeds of those mass spectrum, so we didn't see it. So lots of missing values, and importantly, the missing values are missing informatively. So if you took 645, we talked a little bit about missing data. There's different ways that data can be missing. You hope that it's missing completely at random, which means that the fact that it went missing was like, lightning struck the computer and zapped that one record. Had nothing to do with how sick that person was or anything to do with that could bias the results. Survival analysis is a biostatistics technique where you're studying survival. How long do people survive with cancer? If you want to talk about survival, you have a problem when so-and-so, who you've been observing for 9 months, disappears. They moved to Minnesota or something. I don't know when they died. They lived at least 9 months, but I don't know the exact death time. If not 9 months, I would bias my results if my strategy was just to say, I'll just put in the last time I saw him. So a survival analysis is using statistics to kind of rigorously accommodate this partial information that you have when there's that kind of thing going on. It's called censoring. It's where you have incomplete information about the value. So for a peptide, like let's look at this peptide. I'll be done in 30 seconds. This peptide, while some of them had like 20 observations, this peptide seems to only have like 6, and only one of them was from the control group. The other ones, the chemist would say, are probably down here. They were too low to be detected. Either that peptide was just not present in that rabbit's tissue sample, which is possible, or it was present but at too low a level for the instrument to pick it up. Censoring. It's a censoring problem. These are not missing completely at random. So, all right, questions about that stuff so far? Next time, I will comment a little more about a couple of things that were asked specifically on the discussion board, deconvolution, and I forgot what else was asked for, but in the context of mass spec, we'll do a little more detail on some specialized things on Friday. Okay? Thanks.